my pleasure to introduce Eric Stein, who's our speaker today. I just wanted to highlight that this is a series that we're, I don't know if we can advance this, but we're doing a series of talks with the topic being flows, the targets and ecology. We had our first in the series was held on Monday, and Sarah Yarnell spoke. Eric speaks to us today, and then we have a third in this series that will be on August 18th when Carson Jeffries will be speaking. So I wanted to highlight, I think that the three of these talks are going to really provide us some new insights on how we can think about flows and aquatic ecology. So let me just briefly introduce Eric. He's a principal scientist at the Southern California Coastal Water Research Project, lovingly called SQURP, for those of you who haven't heard the acronym, where he's head of the biology department, and he oversees quite a variety of different kinds of projects, including things related to in-stream and coastal water quality, bioassessment, hydromodification, watershed modeling, and assessment of wetlands and other aquatic resources. And he focuses his research on effects of human activities on the condition of aquatic ecosystems and on developing tools to better assess and manage those effects. Before his time at SQURP, he was senior project manager with the Los Angeles District of the Corps of Engineers. He spent some years with a private consulting company. His background, his bachelor's degree is in biology, with a master's degree very shortly thereafter in science education, and then followed that with a Ph.D. in environmental science and engineering, all from UCLA. So with that, I'd like to introduce Eric and look forward to hearing what you have to say.
Testing. See if we can find it better. Is this one better? No. All right. Yeah. If I stand a little further away. Is that okay? Guys can hear me? Okay. All right. Super. Let me switch my slides over. Got it? Yeah. And desktop. Okay. All right. Yeah. Well, thanks, Cliff. Appreciate the introduction. And so as you heard, this is the second in a series of 3. And so for those of you who were able to hear Sarah's presentation a couple of days ago, I am actually going to recap some of the things that Sarah talked about just to provide some context and continuity, and then also for those of you who aren't able to catch her presentation, it'll give you a little bit of background on work that we're doing at the statewide level to really try to get our minds around this issue of environmental flows. So you can see my really long title, which really has to do with Southern California and aspects of environmental flows that are more focused on some of the water quality implications versus things like fisheries, and I'll talk more about that. But the important point is that all of the 3 talks that you'll be hearing really are part of an integrated framework that all of the sort of logos that you see represented at the bottom and the names on the title slide, this is a group of technical folks that have come together over the last year and a half or so to try to work on figuring out how to make sense of how we approach environmental flows, how do we develop assessment frameworks and tools, and try to inform the variety of different policies in the state that are affected by environmental flow considerations. And these transcend many agencies and many different programs. And so it's an attempt to try to organize the science a little bit to support a variety of different agency programs. And so that's sort of the context that all of this work fits within. So I always start with this, which is sort of like one of my colleagues always says it's sort of like proving that the back end of a pig is made of pork, right? It's the obvious. If you're trying to protect rivers, you have to think, and streams, you have to think about hydrology. And so it is really kind of an integrator, and we know that biological health and the ecological condition of these streams and rivers is ultimately affected by things like geomorphology and temperature and water quality parameters, but all of these are sort of, in a sense, modulated through the hydrologic condition of the stream. And so when we think about management, managing the hydrology of these systems is really fundamental to helping ensure that the ecological condition is what we want it to be. And sometimes that might be reflective of reference conditions, and sometimes that might be reflective of some alternative state that we deem as managers or as society as an appropriate endpoint. But regardless, hydrology is an important element to making sure we're achieving those desired goals. So when we think about a state like California, the challenge is if you sort of think about, okay, this isn't Kansas, not to disparage Kansas, but there's a lot of complexity in the state of California, right? We often talk about the fact that California has sort of continental scale variability in things like rainfall and temperature, elevation, things like that. And so we have very complex environments, and so I've shown you 3 examples of different streams from the state of California, and so how you think about environmental flows for those different types of streams might be very different. And so we have to think about how do we think about environmental flows in a way that's appropriate across different stream types. And then similarly, as we think about the other management concerns and sort of why we are worried about environmental flows, the reasons we're worried about environmental flows might also vary depending on where you are in the state. So in some cases, there might be urban issues such as water reclamation or stormwater capture and reuse. There might be issues associated with dam operations. There might be issues associated with agriculture in terms of either diversion or groundwater use. And so the management issues combined with the different types of streams make it a very complicated issue to try to handle throughout the state. And one of the big challenges, as most of you probably know working in the state of California, is that coordination is always a challenge because the responsibilities are spread among many different agencies and programs, and we don't really have the best mechanisms always in place for coordination between these programs, either on things like data sharing and technical tools or on the policy implementation side. So all of these things make this quite a daunting challenge for a place as complex and diverse as California. And so there is a need for sort of this coordinated framework to try and encapsulate all the things that we've talked about and find a way to sort of work more cooperatively and leverage the efforts, because there's lots of streams and lots of watersheds, and sort of trying to do this one at a time throughout the whole state would take us a long time. And so we want to try to be efficient with the resources that we have. And so if you were attending Sarah's talk, you saw these slides before. The ideas we're trying with this work group is to come up with a framework. We're in the process of developing a framework in coordination with several of the agencies around the state that really talks about how do you think about hydrology and ecology together and use our understanding of hydrology and ecology to set environmental flow targets that can and have a framework that can be applicable throughout the state of California. And then that science piece then can transition into the policy implementation, where we think about things like competing or other uses, beneficial uses, fisheries, and then a lot of the implementation challenges that go with those. And so what I'm going to focus on with the part of my talk today is really more of the technical pieces at the upper part of the figure here. And so the goals are to try and come up with ways to set in-stream flow standards. That would be one goal, but also to assess vulnerability. For example, in Southern California, we're doing work right now looking at climate change predictions on flow and what that means from a vulnerability perspective, and so that's another reason for thinking about environmental flows. We might want to evaluate management actions such as wastewater reuse, or dam operation. And then also when we have instances that we have biological impairment as detected through our existing monitoring programs, often we want to be able to know whether hydrology is the most important management concern or something else like temperature or water quality might be the most important. So for all these reasons, understanding the relationship between flow and ecology is really important for answering all of these questions. And so the framework that we've developed to try and deal with this is to start with sort of a statewide approach, which I'm not going to talk about today because Sarah covered a lot of that. I'll just give you a couple of slides of a recap. But the statewide approach that really is intended to provide sort of a first-tier way to look at how to set environmental flow targets that could be applied anywhere in the state of California at a relatively coarse scale, but then to also provide a framework that says if those coarse scale targets are not sufficient for your application, how do we then have a framework that we can develop more region or location-specific flow targets that take into account some of the very local considerations, and so to sort of tier down into a more detailed type of approach, but do it in a way so that there's enough consistency so that we can share information and we have standardized ways of collecting data so that the information that is developed through these sort of middle tier or second tier programs can be shared. So as an example, yesterday I was in a meeting with the Department of Fish and Wildlife, and they started talking about all this data that they have from different watersheds on fisheries and flows and behavior patterns of salmonids that was completely unavailable to some of the questions that the Water Board was dealing with. So it's sort of trying to kind of find a ways to identify those opportunities and sort of build those bridges. And so again, you saw this, we've already one of the first things that we did through our cooperative work group is we developed this classification system. So we want to try to organize this kind of first tier, this statewide assessment around this concept of hydrologic landscapes. And so we've gone through a process where we've essentially binned or classified the streams in the state into these 9 hydrologic classes based on things like catchment properties, rainfall, geology, and soil. And so these sort of give us an organizational framework to work within for this first tier. I know Sarah asked me to mention that this is currently viewable through the SWAMP, the Surface Water Ambient Monitoring Program Bioassessment website, and we're working right now with the State Board to have this later available in GIS and KML where it can be downloaded. So hopefully that'll happen in the next day or two. And then again, Sarah had talked about that within these 9 hydrologic classes, we can use this concept of dimensionless unit hydrographs to begin to understand reference hydrology, set some preliminary targets based on the reference hydrology, and then also to think about what are the appropriate ecological endpoints that might be relevant for each of these hydrologic classes. And so that's kind of the sort of synopsis or real quick recap of that Tier 1 strategy. And again, Sarah went into that in a lot more detail, so I'm not going to cover it in any more, but I wanted to give you that background and that context for the Tier 1. So the rest of my talk today is going to focus on Tier 2 and give you an example of this sort of more detailed site-specific environmental flows approach, but specific with an application in Southern California where we were not dealing with salmon, right? So, because that's where most of the environmental flows you deal with. So that's going to be the focus of the rest of my comments today. And so just, this is sort of the type of thing that most environmental flow, people who look at environmental flows are most familiar with this type of graphic where we're thinking about a specific taxa like could be a Chinook salmon, for example, and we might have thinking about the life history requirements of this and cues for migration or immigration or breeding requirements. And so thinking about what are the hydrologic conditions that are important to support these life history stages, and then how do we manage the hydrology to support that life history, and how does that affect things like dam and reservoir operation. So that's kind of the majority of environmental flows work that's gone on throughout the state. This is really not relevant for a lot of the issues we're dealing with in Southern California. It's not completely off the table because we do have some steelhead streams, but for the most part, these aren't the driving issues. The driving issues that we're dealing with in Southern California are things like use and reuse of treated effluent. So we have a lot of systems that have been fundamentally modified over the last 100 or 150 years and are driven, the hydrology is primarily driven by treated wastewater or stormwater discharge. And so now as we are in the era of increased recycled water use and reclamation and trying to become more independent of some of the imported water sources, there's a lot of discussion about, well, if we want to reuse some of this water that had been discharged into the streams for other uses, what are the ecological implications of that, and is that acceptable or desirable? We have issues like stormwater retention, and if we're retaining some of those peak flows and trying to use them for groundwater recharge or infiltration or potential reuse, what are the implications of retaining those stormwater runoff flows from the ecological perspective? We also have a lot of concerns about protecting some of the areas that are intact from future land use changes or things like that. And then finally, as we've just come off of our 5 or 6 year drought and we're projecting fundamental shifts in the ecosystems associated with climate change, thinking about how these issues of sort of drought and climate change affect environmental flow requirements. So these are the types of issues that we're dealing with in Southern California. And so fundamentally, what we're trying to do, or what we've been trying to work with in Southern California to set flow targets is create a graphic that looks something, this is sort of a cartoon or a conceptualization of a type of graphic we'd like to create, where we're thinking about on the y-axis, what is the potential change in a biological community of interest. So if we have a biological community of concern, how do we think that biological community might change? What is an acceptable range of change that we can live with? And then how does that relate on the x-axis to some sort of change in flow regime? So if we can create this curve, we can understand how does a change in some hydrologic measure relate to a change in the biological community, and then use that to set management targets. And so for Southern California, we're thinking about sort of streams that are not necessarily big rivers, but sort of in more of those smaller, wadable streams, most of which are intermittent and dry up, or are spatially intermittent or temporally intermittent, so they may not have continuous flow spatially or temporally over the course of the year. The main ecological endpoint that we're focused on are benthic invertebrates, and the reason we're focused on this as our main ecological endpoint is because most of the issues as it relates to environmental flows in Southern California are driven by the water quality programs, not by the water rights or water supply programs. And so the primary indicator we use from a water quality perspective at this point in time are benthic invertebrates, and that's used statewide for all water quality programs, but in Southern California that's the kind of key driver. And then the management issues, as I've already talked about, are things like stormwater capture and water reuse. And so that's kind of the framework for our sort of Tier 2 region-specific environmental flows analysis that we're working with in Southern California. So no dams, no salmon. All right. So then the next challenge is, okay, once we kind of understand kind of the playing field that we're playing on, the next thing we have to think about is what is the analytical approach that we want to use to try to start to tackle this issue of environmental flows. And there's many, many options out there. And part of the statewide framework that I referred to earlier is really one of the things that we're working on there is essentially a literature review that builds off of this document, which was a joint USGS EPA document that came out last year where they summarized existing environmental flow programs throughout the country. So using that, using work that's gone on in Australia and Europe and other places around the world where they've been dealing with these issues, is to think about what are the different approaches out there and how do you begin to select the most appropriate technical approach given the issues that you're dealing with. So given the stream types, the biological endpoints, and the management concerns, what would be the most appropriate assessment tool for you to use? So that's part of what we're working on right now. But in kind of very general terms, these assessment tools fall into sort of two flavors, right, with kind of lots of hybrids in between. So there's like vanilla and chocolate and like a swirl in the middle, right? But the two basic flavors are there's statistical models like the graphic on the top where you're trying to derive a statistical relationship between something like probability of occurrence or density and some sort of measure of hydrology. And then there's more of a mechanistic approach, which is similar to some of the things that Sarah talked about, or trying to think about sort of the ecological history or life history needs of an organism and match that with certain flow requirements. So these are sort of the two kind of flavors that a lot of these methods fall within. So in Southern California, we use an approach called the ecological limits of hydrologic alteration. Excuse me. So we use this method because we think that this is the best of both worlds in a sense. Now I have a crack in my voice. I feel like I'm going through puberty again. All right. So this is a little bit of a hybrid approach because it has mechanistic underpinnings, but is largely a statistical driven approach. And so for Southern California, that was an appropriate choice for us because we're not focused on a specific taxa like a certain bird or a certain amphibian. We're focused on a community. Remember, we're using benthic macroinvertebrates as our assessment endpoint. And so we're really interested in a measure of the entire community as an index. So it's a community-based approach. And the other thing about the ELOHA approach that was attractive for us is we don't want to do this on sort of a reach-by-reach or a catchment-by-catchment basis. We want to try to develop flow targets that are applicable for the entire Southern California region. And so the ELOHA approach is appropriate because it's really well-suited for a community-based analysis versus individual organisms, and it's also well-suited for kind of a regional versus a location-specific type of analysis. And so the basic steps of this ELOHA approach are that you have to estimate the degree of hydrologic alteration. So the basic premise is you're relating hydrologic change to biologic change, so a delta H or to a delta B, delta hydrology to delta biology. So that means we have to be able to estimate the degree of hydrologic change between sort of a natural and current conditions. We want to compare that hydrologic change to some sort of response in the biological community. then we're going to use that relationship, that sort of conceptual curve I showed you, to develop some sort of indices or scores that we can use to assess and prioritize how we manage the flow in different streams, and then we're going to apply that to evaluate management actions and set management targets. So that's the general approach that we're going to use. And so this graphic just basically, again, shows you that the steps we went through is we started with that statewide classification that I showed you. We're going to compile our bioassessment data, estimate hydrologic change, compare the two, select the metrics, and then come into management. So that's the process we went through. So the data set, we're fortunate in California because California has a robust bioassessment and biomonitoring program, and so there's been something like close to 5,000 sites that have been evaluated over the last 15 years or so using our traditional Benthic macroinvertebrate data set. This is the Southern California portion of the statewide data set. There's about 800 sites that we've assessed in the last 7 or 8 years. So that was the biological data set that we had to work with in terms of understanding kind of the biological condition using benthic macroinvertebrates. The problem is that we need to be able to estimate hydrologic change, or the delta H part of that graph. We need to be able to estimate hydrologic change everywhere where we do bioassessment. So we have 800 sites. We need to estimate some degree of hydrologic change at all 800 of those sites, and as you're probably aware, very few of them are gauged. So the first challenge in this approach was how do we deal with this issue of estimating hydrologic change across this broad array of sites in a parsimonious manner. So the way we did it, of course, when you don't have data is you go to models, right? And so that's possible, but we still don't really have the resources or the time to develop 800 models. That's a lot of modeling, or a lot of modelers, depending how you use it. And so what we decided to do is go with what we call an ensemble model approach. And so the ensemble model approach basically means that what we're going to do is we're going to create a subset of models. We're going to pick a subset of models that are representative of the range of watershed conditions that we have in Southern California. So some large, some small, some steep, some not so steep, some higher rainfall, some lower rainfall. So we want to encapsulate kind of a range of watershed types and pick a representative set of models, then calibrate and validate those models with the data that we have. And so now we have essentially what amounts to a library of models to choose from. And then we develop what I called here a model assignment tool, which is essentially a statistical tool that allows us to go to any new watershed, any new catchment of interest. So if we have a bioassessment site and we delineate the catchment draining to that, I can calculate some basic properties of that catchment, like size and slope and things like that. And then based on the physical properties of that watershed, I can pick the right model out of my library or out of my ensemble, pick the correct model, tune very few, about 3 or 4 different parameters get adjusted in that model, and then I can run it. So it basically allows me to sort of create this library that then I can, in a very parsimonious way, apply across a broad range of sites in a fairly efficient manner. And so in our case, we picked 26 models, and so these were at gauged locations. You can see we have a range of catchment sizes, a range of elevations, a range of impervious covers, and then we calibrated and validated these models with field data, and then we created this clustering algorithm using basically it's a variation of a boosted regression tree analysis to essentially create this binning that allows us to go to any new site, select the right model from our library, and assign it to our site of interest. And so then the way that looks is if we have some sites of interest where we have bioassessment data, like shown here on the map with the red dots, I can use my model assignment tool. I can pick the right model off the shelf. I basically run it under current land use conditions, and so that gives me a flow series, a daily flow series that's shown in yellow here. That would be sort of the current hydrology. So we ran essentially a 6-year simulation for every one of those bioassessment sites. We run a 6-year simulation. It's a synthetic simulation that includes wet, dry, and average conditions. So we've sort of encapsulated the range of conditions. And so I get a measure of sort of the current daily flow over sort of a 6-year period that represents a range of hydrologic conditions. Then I go into the model and I turn that catchment natural, which means I make the land use natural, I change some of the storage parameters in the model, I change a couple of the routing parameters in the model, and then I rerun the model using that same 6-year rainfall record, so the exact same precipitation record, synthetic record into the model, run it under natural conditions, and then I get the blue hydrograph. So now for each of my 800 sites, what I essentially have is reference hydrology and current hydrology from the model. So again, this is not measured, it's modeled, but I do have now some way to compare current to reference hydrology for each of those 800 sites, and that allows me essentially to calculate that hydrologic change. So the delta H, or the hydrologic change, is essentially just the difference between those two hydrographs, right? Simple, pretty straightforward. The next challenge is, that's great, but I have now 6 years of daily data to compare. And so usually what we do in this case is we want to create some sort of flow metric or some sort of measure that sort of aggregates different elements of the flow. And so if you go into the literature and look at flow metrics that have been used over the last, 6 or 10 years, there's literally hundreds of different flow metrics that people have published on. There's standard packages like the Index of Hydrologic Alteration that have been used. But in a sense, these metrics can fall into these basic categories. There's metrics that relate to the magnitude of flow, the variability of flow, the duration of flow, and the timing of flow. And so we wanted to have some metrics that we looked at that were representative of all those different elements of the hydrograph. So we're not just capturing magnitude, but different elements of the hydrograph. So we compiled about 300 of the metrics that we felt sort of represented the range of these different elements of the hydrology and were things that had been previously published on. And then we calculated those metrics using, we went into those hydrographs that I just showed you. Now we have essentially current and reference hydrology, and so in an R package, we can just churn out those metrics for every one of those 800 bioassessment sites for both current and reference conditions, and then we bin them by average, wet, dry, and all year. So now we have the ability to look at the overall hydrology and then under different climatic conditions. So essentially now what we have is a set of metrics that speak to hydrologic change. So we have change in magnitude, change in variability, change in duration, change in timing for all of those 800 sites under different climatic conditions. So lots of hydrology data. So the next step was then, how do we know which ones are important, which ones to use relative to our biology? And so again, this is largely based on a statistical analysis. We basically created a bunch of curves that look like this. This is a logistic regression curve. And basically what we're asking is, on the y-axis, we're asking what is the probability of change in biology, and on the y-axis, we're looking at the change in hydrology. So this is that same curve I showed you at the beginning, change in biology, change in hydrology, except now I'm just showing it to you with real data. So let me talk for a moment about the y-axis, because what we use, I mentioned that benthic macroinvertebrates is what we use as our ecological endpoint, but the measure that we use is something called the California Stream Condition Index. So I'm not sure how many of you are familiar with the California Stream Condition Index, but it's sort of like an index of biotic integrity, same concept. It's just it's a single index that aggregates measures of biological community health. But the important thing to know about the California Stream Condition Index, which is the standard tool we use in California, is that it's what we call a predictive index. So what that means is the way you calculate the California Stream Condition Index is you basically pick a site and you simulate what would the biology at that site look like under natural conditions. So that creates what's called an E, or an expected value, right? And you create an expected value for that site, and then you go out and measure the bugs like we normally do, and you see what's there, and you compare the observed to that expected value, and the ratio of the two gives you that CSCI score. So it always goes from 0 to 1, with 1 being the site matches expectations under reference conditions. So the reason is that that's important for this analysis is because that essentially is a delta B, or biologic change, right? Because it's what is my current condition relative to a reference expectation. So what I've done is I use a CSCI, which gives me a measure of biologic change from reference on the y-axis, and then on the x-axis I have my measures of hydrologic change that I just talked about. So now I can create that graph of delta biology versus delta hydrology. So that's essentially the analysis we used. The reason we use logistic regression analysis is is because the y-axis is reflected as a probability of change. And so as a scientist, I love that because I don't actually have to make a decision what the right number is. I don't have to say, well, this is the target, because what it's telling me is I pick a certain threshold. We have an established threshold in the literature for the CSCI. It's 0.79, and that number is sort of the break point between reference and non-reference conditions. Now, that number doesn't have it's not a regulatory blessed number. It's just based on comparison to the distribution of reference sites throughout the state of California. So 0.79, above that we consider good sites, below that we consider not so good sites. And so what the y-axis here shows is what's the probability that my CSCI score would be basically above or good, above that 0.79 value. And so what I'm showing you in this graph is basically the 50% probability. So I can tell you here for a certain hydrologic metric, which is here is high duration flow days, and this is a decrease in the high duration flow days. So for if I want to make sure, if I want sort of a 50% likelihood that I'm going to have good biology in my streams, I want to make sure that my high flow duration does not decline by more than about 6 or 7 days. So that's basically the way you look at this graph. So basically at a 50% probability, I come down onto the x-axis, so it's a minus 6 days. So if I have anything, minus 6 days or less, then I'm going to be in good shape in terms of my biology, or I have a 50% probability. Now, you as a manager might decide 50% isn't the right number. I want 75%. Well, that's fine. Just go up to 75% on the y-axis, go across, and pick a different number. All right, so that's the way these logistical regression plots work. So we can generate these for all of our biology, hydrology combinations. And again, just to make it more complicated, the shape of this curve in the decreasing direction and the increasing direction, the shape of that curve can be different. So we have to look at those independently. All right, so lots and lots of data. And so we produce lots and lots of these plots. So we have lots and lots of these plots with different shapes. And then we essentially go through a selection process where we basically go through all of those plots and we pick the ones that have the strongest relationships with the biology. And then we run them through another set of filters. We want to make sure that the metrics can differentiate reference from non-reference. So if they can't, that's not very useful for us. We eliminate redundant metrics, so we want to have metrics that are non-redundant with each other. And then we want to have metrics that are amenable to management, meaning you can actually do something from a management perspective to influence that element of the hydrology. So after we run it through all those filters, we wind up with a set of metrics, a table that looks like this. And so this is kind of our final set of metrics that we produced, where we looked at a series of, in this case, 7 metrics. And we identified some, in some cases, some increasing or some thresholds in either the increasing direction or the decreasing direction, and we identified the critical precipitation condition. And so now you can use a matrix like this to create sort of a management template to say, if I can manage a stream within these certain different parameters, then I can have, in my case, a 50% probability of having good biology in that stream. And so the important thing about the thresholds is they're not absolute, is they're relative to some sort of baseline condition. So they can be tailored for whatever stream you happen to be interested in. All right. We then aggregate, so those are the individual metrics. We just did a simple adding them all up and then bin them into these A, B, C, and D categories. And so now I can go back to my map that I showed you at the beginning of all the bioassessment sites. And for each of those bioassessment sites, I can put them into a category of hydrologic condition based on whether the hydrology, the current hydrology, has deviated from reference condition to a point where we think it's going to create a hydrologic alteration. And then I can color code them with blue being good and red being bad and a few gradations in between. So the first step, it allows us to kind of get a sense of kind of the hydrologic landscape within the region. But what we really want to do is use this for management. So I'm going to give you a few slides of some management applications. So one of the ways we've used this is we did this in the San Diego River Basin down in San Diego County, and we went in and we looked at the current hydrologic condition using those same categories under the current land use, 2016. We overlay the county's general plan, what their proposed development scenario is like for 2050, and then we can project where we think the hydrologic conditions as they relate to biology. So this is hydrology as it's the type of hydrology that the bugs care about, essentially. And we can show where in the watershed we think conditions are going to be getting worse, or which areas are at risk from a biological condition perspective. If you allow that land use to go forward and you don't mitigate those hydrologic impacts through BMPs or other things like that, where do we think the areas that are going to be at highest risk? And so you can use that to prioritize management decisions. We can also go in, and this is the same watershed, and we just created this simple matrix where we just ask the question, where is the hydrology good or bad, and where is the biology good or bad? And so we created this little sort of contingency table at the bottom, and we can then look at that and say, well, in areas where my biology is poor, it's below that 0.79 value I told you about, and my hydrology is also poor, those are areas where I want to prioritize my flow management actions because I've got poor biology and poor hydrology co-occurring. In contrast, if I have poor hydrology or poor biology and good hydrology, which is other stressors, that may be a case where maybe hydrology isn't the most important factor, maybe there's some other stressor at play there. And so we can use this to prioritize management actions. And the last thing we can do with this is we can use this for scenario analysis. So this is, again, one step down beyond just kind of characterization. This is the Alvarado Creek subwatershed within the San Diego River. And we basically, in that case, this is an area that's already built out. The current effective impervious cover for that built-out watershed is about 50%. And what we want to do is we want to try to manage that hydrology down to a level where we think it's going to be protective of the in-stream biology. And so we evaluated a series of different BMP implementation scenarios that changed the effective impervious to either 5, 10, or 25%, as well as looking at the 85th percent capture, which is what's in their current permit, and said, well, how do we think that's going to change the hydrology and will it affect the biology? And so what you can see here is the model that shows the current hydrology and what it would look like under 2% impervious cover, which is what we consider reference conditions. And so this table essentially picks out a few of those flow metrics that I showed you, and it really just shows what the target is in the far right, and then it shows you under the 2, 5, 10, and 25% impervious cover BMP scenarios, how would those metrics perform relative to the target? And so they're color-coded. If they're in green, it means you've met your target. And so you can start to look at these types of different scenarios and say, how much BMP implementation do I need to put in order to manage the hydrology down to a level where it's going to create those healthy bioassessment scores. And then we also looked at the 85th percent capture, which in this case was probably the best performing BMP option. All right. So that's kind of just an example now of walking through the process and then showing you some applications of the process. And so as an output of this, we have a series of data products which we've made available, the GIS for the stream classification, all of those hydrologic models, the ensemble of models, and the model selection tool so they can be used for other purposes, and then all of the biological models that I showed you, those are all available so that we can continue to build and use those for other regions. In terms of where we're going next is we're continuing to do work I told you about. We've done this for the benthic invertebrates. We also use benthic algae as another one of our bioindicators, and so we're starting to develop or have some initial plans to develop this same type of relationship for algae. We're doing some preliminary work looking at some of our local fish and some of our general riparian habitat using some different models. Additional causal assessment work to improve our ability to discern flow versus habitat. So this is some of the work that Carson is going to touch on when he comes, I think, next week to talk about sort of how you bring geomorphology into this and start to tease out some of those physical habitat effects. One of the things that we're particularly interested in is this concept of shifting baselines. We know that the flow regimes of our streams are shifting due to climate change and changing water use practices, so we have to think about how this sort of slow, gradual change in the hydrologic regime might affect the biology. And then ultimately, as I started the talk with, is to integrate this concept into the statewide overall framework that I started the presentation with. And so with that, I will wrap up and hopefully have some time for questions.
Are there any questions? you explained this really well, so thank you. So my question is, obviously you're comparing the hydrology with the biology here. Have you begun to integrate sort of the other beneficial uses, such as water supply reliability at the local level, or in a way that your second to last slide already indicated this, on rainwater capture, where you can add other models, predictive models or scenario models to the two functions or the two pieces that you're comparing now. Right. Have you started to integrate that with what's going on the LID front, or rainwater capture and water supply reliability side?
Yeah, no, it's a good question. So we haven't done a lot on that. We're just, I alluded to this a couple of times, we're starting to do some preliminary work with the LA Regional Board, and they have sort of two questions. One of them is sort of a climate change and drought preparedness type of question, is their main question. So as they're looking at water management first in the near term as it relates to drought preparedness, and then in the longer term in anticipation of future climate change, and asking how do we balance the need to manage water differently with the potential implications for the organisms that live there. And so we'll probably use some different models. Probably the most obvious example is one I showed at the beginning, is treated wastewater effluent. And so as we start to reuse that a lot more, what are the implications of not allowing that water into the stream and using it for other beneficial uses potentially relative to the in-stream ecology. And so we're just, I would say, at the very, very beginning of starting to think about that. And I think, as you alluded to also, it requires us to bring in other modeling approaches and other sort of tools to get at those really complex interactions. So, yeah, it's a really good question.
Eric, my question, well, I actually have two questions, but the primary one is just, can you list a few of the entities? You mentioned the regional boards, but who are actually being able to use some of your findings, first off, and then secondly, I'm kind of curious. You mentioned the effects of water not going into streams from stormwater capture and so forth, but I'm kind of curious because I've always thought of, has anybody ever looked at the implications of adding the state water to the Santa Clara River watershed? The hydrologic effects.
Yeah.
Or biological effects there.
Right. So there's a couple of questions there. So the first question had to do with the entities, the sort of agency entities. So in Southern California, most of the folks we worked with have been the regional boards and the local municipalities. So the regional boards on the regulatory side, and the municipalities who are the ones, the sort of management entity. A little bit with the Department of Fish and Wildlife, but those are the main folks. Statewide, we're basically 4 entities we've been working with, primarily statewide, which are Department of Fish and Wildlife and their in-stream flows program, DWR, and then there are 2 parts of the Water Board that we've been working with. One is the Division of Water Rights, who has a process that they're going on to develop regional in-stream flow evaluation methods, and then the Division of Water Quality, which is sort of more focused on some of the things I talked about. So those are kind of the main, I would say, entities that we've been interacting with. In terms of your second question, yeah, so the hydrologic change going forward when you look at it, can be too much. It can be an augmentation or a depletion, essentially. And so that's why I alluded to this when I showed the logistic regression curve, as we split those at sort of the 0 point, and we generate one of those analyses for both the decreasing direction and the increasing direction, because we want to try to understand biologically, do we see more of a relationship in sort of a declining where you have less water, or an augmentation where you have maybe more water, and the patterns and the shapes of those curves can be quite different. And so we're trying to get a little bit of understanding of in which direction is there more sensitivity. And so that's why on that table you saw in some cases there were 2 columns on the right. One said increasing thresholds in the increasing direction, and one said thresholds in the decreasing direction. We'll back up to that. And in some cases, we have thresholds for just one versus the other, some cases both. And it really, in this case, it really depends on where we had the strongest response in the biological community relative to that hydrologic change. I know we had a question online also.
Yeah, let me go see if we could find that online question.
Okay. Okay, so the online question says, where can we access the data products, GIS layers, hydro, and biomodels that you've made available? So right now, so there's a couple of different answers. So the GIS layers, so like, for example, the stream classification, in fact, I emailed the State Water Board person this morning to ask them if it was up yet. As I said, I saw it up about a week ago, and I checked this morning, and it wasn't up. So the idea in the short term is that those GIS layers would be available through the SWAMP Bioassessment webpage as part of the data downloads there. We do have a page, we have an environmental flows page right now on the California Open Data Portal, which is data.ca.gov. And if you go under water, there's an environmental flows page. And so all of our products from Southern California, the hydrologic models, the biologic outputs, all of that is available on data.ca.gov, so you can get all of that there. And then ultimately, our hope is we're hoping to establish a portal through the California Water Quality Monitoring Council is our hope that we would be able to use as another home to put a lot of these things that not only we're doing, but sort of the larger community of scientists and practitioners. But in the short term, the stuff that I showed today, that's where you can get it. And if you have any problems, you can just email me, and I can always send you the links. So.
Thanks. This is very interesting. I have a couple of questions. I spend a lot of time working with geologists who work on these types of systems, and I assume that most of your systems are spatially intermittent.
Right. Yeah.
Is that correct?
Yes.
To what extent, if you overlaid lithology or if you overlaid tectonics, would you be able to locate where many of these perennial segments belong?
Yeah. Okay, yeah, I'll repeat the question. So the question is, if I overlay geology or lithology, could I get a better handle on sort of the perennial versus non-perennial segments of the stream? So again, really good question. I think that was not something that we specifically looked at through this project. I think, but it is a really good idea. But I will say that there's other efforts going on in the state right now. There's an effort to try and this is an effort that started with some work that USGS has done out of the folks back in Reston, Virginia, and we've sort of continued it here in California with some funding from the water boards, but it's attempting to do exactly what you said, bring in rainfall, geology, soil properties, elevation, things like that, and sort of ask the question, can we create models that predict likelihood of perenniality? And so as we know, perennial and non-perennial is not binary, right? It's sort of a continuum. And so what we do have is we have a preliminary data set that was produced statewide by USGS. I think we're working on trying to get it available online because it's a pretty big data set. But what that data set, this was work that Darren Carlisle led up. And what he did is he developed some models throughout the state, statistical models. And for he used the NHD plus data set, so the stream network. And so for any location on that, any one of the reaches on there, he predicts the probability of greater than 0 mean monthly flow for each month of the year under average wet and dry conditions. So it's a big data set. But what it allows you to do is go into a certain reach and say, what's the probability of September flow being greater than 0 under wet conditions? Or what's the probability? I have a graphic of that in a different PowerPoint. But the idea is that it begins to get at that question of what's the likelihood of seeing flow, at this reach under these climatic conditions. So I think it's a really great product because it's statewide and it helps us start to get at that. It's clearly, it's not perfect, but it's, I think, way better than what we get out of the current NHD layers. And we've been starting to use it to inform, like, a lot of our bioassessment programs when we have to go and, select sites for assessment. We want to try to avoid sites that we think are not going to have water in them. So. Are there any other questions?
Thank you. Appreciate it.
I don't know if there's another question online, so if people are still online that says, I'll just read it. Norman Johnson says, seems like you have natural current change for hydrology, but I'm not sure how you tease out the same delta for biology. So I'll just, I touched on that briefly, but again, that was the whole point of the CSCI. The CSCI, by definition, is a delta biology because it's a measure of observed biology relative to expected reference conditions for biology. And so by definition, the CSCI is delta biology because it's observed relative to reference. And so that's how we get at the delta biology piece.